This is lecture 15, and the main focus is the plant hormone auxin, which has a huge role in development. We're going to talk about the different kinds of genes that affect the production, the transport, the movement around the plant, and the response, transcriptional response to auxin during development. And I will cover a small number of mutations in particular genes that affect development that are related to auxin. In a couple of cases towards the end, I will just talk a little bit, introduce sort of shoot and root meristems, and a couple of the genes that are responsible for setting those aside. We're going to revisit this topic both when we talk about leaves in the next lecture, the placement and spacing of leaves and how those are specified, and you will hear a little bit about some of them again when we talk about the decision to change a regular shoot into a floral shoot so that it's going to produce flowers and so on. And there's a set of learning outcomes that I put in your post-it notes, but I decided not to. I'm always worried these are going to go way too long, and so I didn't put the learning outcomes in because that's something you can read for yourself. So auxin, there's a number of, those of you who take plant biology will learn a ton about different plant hormones. There's lots of them, and they're interesting. But auxin, or indole acetic acid, is one of the major ones in development. And the genes that mediate the response to auxin. Auxin itself, as you know from last lecture, isn't a transcription factor. It basically lifts the inhibition on the transcription factors that then go ahead and transcribe the genes. And auxin plays roles in the elongation of cells. You can reshape, you can cause a stem or a root to bend by lengthening the cells on one side and leaving them the same size on the other side, and that will cause a bending towards the shorter side. Regeneration of some conductive tissue after wounding. Apical dominance, I think I mentioned in the previous lecture that the most apical bud is usually dominant. If you remove it, though, when you pinch back a plant, for example, to make it bushier, apical dominance is lost because the bud is gone, and so the lateral buds start taking over. Both photo and gravitropism, which I'm going to talk about briefly. Adventitious roots, so these are new roots that form, and if you've ever taken a cutting of a plant, a lot of us have done that, you clip the stem and then new roots form at the node, which is the place where the leaf emerges. If you remove that leaf, you can even just, for a lot of plants, just put them in some water and get new roots. And fruit development, which we aren't going to really focus on, but that is one of the things that auxin does. So in 1926, Fritz Wendt, or Wendt probably, just kind of started some work on this, and he basically had shoot tips. I can't remember what the organism, if he was working with corn or something, or wheat, I can't remember. But basically he decapitated the little plants. He cut the little tips right off of the growing shoot. And then he took that shoot and he put the tip, and he put that on a block of agar. No, after that growing tip was removed, the shoot didn't continue to grow. But what he found was that if you took the tip and put it on some agar, just sort of a gel-like material, and allowed some time, something from the tip sort of entered, diffused into this agar block, and you could take the block itself and you could set it on the shoot and cause that shoot to start growing again. So there's something in that shoot that allows for the growth to continue, right? And if they're well centered, the shoot continues to just grow straight, up towards the light. But if you offset, so if you put the block only on part of the shoot, then when the shoot started to grow, you basically had longer cells on the side where the hormone was, and shorter cells on the opposite side. And so that causes, if you think about this, these cells are getting longer, so they're pushing up more, but these guys are shorter. You're going to get this bending of the tissue. And we won't go into all the details of all the experiments, but they finally were able to isolate the hormone auxin and demonstrate that that was the substance in those shoots that was responsible for that behavior, the ability for the shoot to continue to grow and the bending of the shoot if you didn't set the block symmetrically on the shoot. So plants exhibit what is called positive phototropism. So a tropism is a growth towards or away from, or a movement towards or away from a stimulus. So plants tend to grow towards light. Oops. And if the light is coming from a particular direction, it turns out that on the shaded side, so the side that's getting less light, you have an increased concentration of auxin. And that causes the lengthening of the cells. And when you lengthen those cells, they're going to grow more, but these guys are anchored. They're shorter, and so you're going to get an overall bending. So the long side is on the outside of the bend, and the short side is on the inside of the bend. And plants also respond to gravity. So the stems, the shoots, grow up and away from the source of gravity. So this is called negative gravitropism, sometimes also referred to as geotropism, towards the earth. And then roots will grow down towards the source of gravity. So that's positive gravitropism. So you want to think about this question. It may come up. If auxin is causing lengthening of cells, then where in this shoot is the auxin located? And where in the root is the auxin located? It's interesting that the two different parts of the plant have these different responses to gravity. Okay, so what kinds of genes affect the production of and response to auxin? Well, the synthesis genes. So these are genes required to produce the hormone auxin. And those of you who took 357 will know these categories already because we did our CRISPR constructs on genes like this, but we didn't talk very much at all about the biology of them. So here's where you get to learn a bit more about them. So there are certain genes that encode enzymes that are required to produce the hormone. There are a number of proteins that are responsible for moving auxin around. So it turns out that polar transport, so transport towards one end of the embryo or the other, towards localized regions within a developing embryo and so on, is really important for development. So the transport is essential. If you lose the ability to move the auxin around in the embryo, you won't get proper development and then response. The transcription factors and some of the proteins that control those that are responsible for regulating which genes get expressed. Now the synthesis and transport proteins, there's a small number of these, and most of them are, pretty much the same in all cells. This is not 100% true. There are some transport proteins that are found in one cell type, but in general, the responses will be the same in all cells. Whereas the responses, different parts of the embryo have a different set of target genes that they might transcribe or repress in response to auxin signaling. And so these are going to be quite different in different parts of the embryo. And this is just an example. So there are mutations, a whole series of them called PIN mutants. And they have this funny needle-like phenotype. So this is one that actually affects the floral, the inflorescence. So this is the stem that gives rise to the flowers. And when you look at it, here's, they're called pin for obvious reasons. They just look like these long, skinny structures, and they haven't developed into the correct structure. And so there's a whole bunch of these. There's AUX and there's PIN mutants, and in both cases they are responsible for the transport, and they're paired, and we'll talk about that in a couple of minutes. So there's other phenotypes that you see. If you've got mutants that don't produce enough auxin, you can see mutant phenotypes that look very similar to this. So how can we tell which is which? Well, just think about that, and we'll get to it in a minute. So I thought I had a, yes. So just think back to this slide here. And then, so how do we tell the difference? So the AUX and the PIN mutants don't respond if we add auxin to them. They already have plenty of auxin, but they just can't move it around to where it's supposed to go. So you cannot fix this or quote unquote rescue this if you give them auxin. And the AUX genes are influx carriers. That means they move auxin into a cell. And the PIN genes encode efflux carriers, which are moving auxin out of cells. And if you think about the cells of plants, in order to get auxin from one cell to the next, you first have to put it out of, so the PIN is first, but then it has to be taken into the next cell. It's like, think of it as the baton being passed. You've got one person passing the baton away from themselves, and then the other one is grabbing the baton and taking it, right? And then the ARFs, the auxin response factors, these are transcription factors and some other regulators. They also don't respond if you add auxin, because they already have it. There's nothing wrong with the auxin that they have, but they're not capable. The transcription factors are mutated, so they can't respond to it correctly. But in contrast, the actual mutations involved in the synthesis can be rescued by treating with auxin. So you've provided the substance that's missing, and now the development can be fixed. Okay, so these kinds of experiments are one of the ways you can determine what kind of gene is mutated in plants that have these auxin related phenotypes, because the phenotypes look very similar. If you can't make the auxin, if you can't transport it to the right place, and you can't respond to it, you're going to have the same phenotype, and we can distinguish between them experimentally a little bit with this. And then of course there's more experiments that have to be done. So the very first cell division in the embryo, where you have the apical cell and the basal cell, auxin is part of establishing that apical identity of the embryo. And so there's auxin produced in that basal cell, and it's transported up into the apical cell. And in a second, we'll look at a couple of the genes that are also responsible for further, excuse me, further expression, further development. Once the embryo has developed a little bit, and you've got this proembryo, which is the little embryo proper, and this is the supporting tissue, the basal cell and the suspensor, the auxin is being produced, and it's being sent up into the embryo proper, the proembryo. And not only is it being transported into these cells up top, but there's the cells up here are actually transporting it back and forth to each other here. And the idea is that all the auxin is winding up here. So you have this, what we call local concentration, and it's often referred to as an accumulation as well. Okay, I'm not that concerned whether you memorize these, but to know that it's PIN proteins which are moving, are responsible for moving it in here. Okay, a lot of the AUX ones aren't listed, so these are the ones that are going to be mentioned for the most part. So this is how the first apical and basal axis is specified. And now this is from a publication, and it just has a little bit more information. So this is at the early cell stage, and there's just 2 genes that you need to know. There are, yes, a bunch of other ones, but so these are called WOX genes, WOX2 and WOX8. And so the idea is just that when you look at like your embryo, the zygote at first, you can see that the proteins are sort of everywhere there, But after that first division, where the embryo is one cell and then you've got the suspensor down here, now you have WOX9 and WOX8, and we're only going to focus on WOX8 in the basal cell, but WOX2 is expressed only in the apical cell. Okay. And that's continued through. So don't worry about the details of it, except just to point out that the WOX genes are asymmetrically distributed, and they work together to maintain the apical basal sorry, polarity. And it is and one of their roles is basically promoting, modifying the actual the pin and OX protein, so that the transport is effected in just one direction only. And so this is I like diagrams something like this that kind of helps me remember just the basics. So as I said, this is oversimplified, and you can see from the experimental work previous it is. But a diagram is really helpful. So this is our zygote. It's got WOX2 and WOX8. But after that first division, the WOX2 is all up top in the apical cell, and the WOX8 is in the basal cell. And if you look at mutations in WOX8, what happens is that WOX2 is kind of lost in that apical cell, and the correct movement of auxin in that and other cells is also lost. And so this tells us that these genes are required for the auxin gradient. WOX8 is required for WOX2's expression, and together there seems to be a regulation of especially the PIN gene expression. So they're responsible for activating and making sure that the PIN, the transporter proteins, are present and correctly localized, so that our auxin is going to go up towards the apical cell and the cells after that for a little bit. Okay, so there's a couple of other genes that I talk about a little bit, and if I talk about sort of asymmetric distribution of particular proteins and so on, making yourself a little diagram like this can really be helpful in understanding how that works. Okay, so that's a little bit of detail on this this very first diagram here, which is, as you can see, horribly oversimplified compared to the real experimental work. And this too, but this, like, if you remember this, then that's going to be good enough from my perspective. But then when we get to, this is about the 32 cell stage approximately, and this is the early globular stage embryo. And now the auxin isn't being made in the basal cells anymore. It's being made in the apical-most cells of this stage, and the transport is all down towards the hypophysis. And now you've got a new place where it's localizing. So the first step is to sort of specify the apical end and the part of the early embryo, early structure at least, that's going to form the actual embryo part. And now it gets transported the other way down to the basal part of the embryo, not all the way down to here, to specify the tissue that's going to form future roots and the root meristem in this region. Okay, so I've been talking about efflux and influx carriers, and I just want to point out that if you're going to move auxin from one region to another, especially because you wind up with these local concentrations. So what you're doing is you're moving the auxin towards the higher concentration, and that means it can't happen passively. It requires energy. So what you've got are these little, special transporters that the efflux ones move the auxin out, and then the paired ones on the other side pick it up and transport it into the cell. And that requires energy because you're moving it against its concentration gradient. If you were just going to let it diffuse, it would want to go from the region of higher to lower concentration, right? So that's how we can tell it's an energy requiring process. And so the transport direction changes multiple times during development, and the way you change the direction is you have to change where the transporters are in a cell. So there are actin filaments that carry them to exactly where they need to go, and then they're put into the membrane through an exocytosis process. And then when it's time to take them away from where they are endocytosed, attached to an actin filament, and then moved to the new place. So if we wanted to move the direction of these guys would have to go to that surface, and these guys would have to be, so if we just called this right and left, just for the sake of argument, left, right. So here you'd have the efflux transporters on the right side of the cells and the influx on the left side so that they can be paired. But if we wanted to change the direction, these guys would have to be over here, picking up and bringing in from the opposite direction, and these guys would be down here sending it out, like remembering that, you know, that there's extra cells, right? It's not just a two-cell thing. And so it's beautifully coordinated because the cells can reasonably quickly change the direction of transport by pulling all these guys in through endocytosis, sending them along the actin filaments to the new location, and then exocytosing them out onto the membrane, not the cell wall, but the membrane of another face of the cell. Okay, and then I just want to talk about a few mutants. So monopterous, monopteros, so it basically means one leaf, and that's because if you looked at an early little seedling of an Arabidopsis, it has these two cotyledons. That's what these two things are here. And the monopterous mutant has apparently a fused one. And basically, they don't have any hypocotyl. They don't have any root meristem or anything like that. And that is because the embryos haven't got the ability to respond to auxin transported to that region. And so it's an auxin response factor. And this is another picture of it where the flowers aren't forming. These aren't complete loss of function, they're weakly active, but basically there's plenty of auxin being moved in this this plant stem, but it's not able to produce the flowers it's supposed to. Because why? Because the monopteros is not able to transcribe the correct genes needed to form the flowers. There's another one here, embryo mutant 4. I don't know why this S is here, for topless. So there's a gene called topless. We're talking about meristems now. So we looked at the root meristem, and this gene is needed normally in the apical half of the embryo. But when you make a mutation in the embryo, they don't have any apical basical polarity. So instead of the apical meristem developing as a shoot, it develops as if it's a root. And so you have roots developing at both ends of the embryo instead of shoots here and then roots at this end, right? And so just to this mutant embryo is being stained for a gene that's specifically expressed only in roots, in the developing root meristem. And so you can see the normal expression here, and then the mutant has abnormal expression here. And so you might want to make yourself a diagram or chart similar to that WOX one that I showed. Topless protein normally functions in the apical half of the embryo. It forms a complex with OX-IAA proteins. and it acts along with them to repress the auxin responsive genes that are required for root formation. So up in the shoot apical meristem, we don't want roots, right? And so we have to shut those ones off, and that's what topless does. But when we have a topless mutant with the loss of function, the phenotype is that the root apical meristem forms both in the apical and the basal parts. So you don't have a shoot apical meristem at all. It's replaced by the root apical meristem. So use these blue questions as prompts for how to think about this material. So if this is what we know that protein does, what is it doing? What is its job? And then if it's mutated, how does that phenotype fit in with the job that it's supposed to be doing, that it isn't? Okay? And the meristems, I know that in the previous prep, I showed some images from the text that basically, show that the embryo, there's fate maps that you can draw starting in that torpedo stage. But it turns out that the meristems for the shoot and the root have already got specific genes being expressed in them, and you can actually stain for those and see that the shoot and root apical meristems are actually being set aside earlier than the torpedo stage. Okay. Now, meristem tissue is really interesting because it's what we would call pluripotent tissue, and it's stem cell tissue that goes on for the whole life of the plant, right? So this could be a giant tree, and it could be 3,000 years old, and it could still have meristems and start developing new leaves, new shoots. It could still make flowers and fruit if it's a fruit bearing tree, and so on. And if the plant has damage to the roots, it can form new roots as well. So these are really cool tissues, and it'd be useful to have such tissues in people, so that we could replace body parts, for example, if anything was damaged, etc. So STM is a gene called shoot meristemless, so that's the name of the mutant phenotype. It's normally expressed right in that presumptive meristem tissue at the heart stage. And if you make a mutation that inactivates that gene, then you don't have the meristem tissue. It's no longer a self-renewing tissue. So what happens is in the mutant plant, germination occurs, and there's the cotyledons, and a couple of abnormal leaves form because some cells have been set aside. But then what happens is they're not, once those few cells that have the capacity to form leaves are used up and form the leaves, there's no self-renewal of the stem cells, in the meristem. And so it just runs out, and you've just got, there's nothing here. There's no further development. You've got this funny, very defective looking plant. Okay, so this is the mutant. Now you can also do things like you can overexpress them. So what happens if you turn on STM and you overexpress it? Then the whole embryo is just a whole bunch of cotyledons and little presumptive leaves, and you don't have any proper stem, you don't have the leaves forming properly. So, this is too much STM gene, and this is too little, right? So when you see the lack of the gene and too much of the gene, that tells you what the normal gene is supposed to be doing. Okay, so these are the genes to know. And this tells you what their relationship is to auxin for these ones. And these guys down here we'll be talking about again in the upcoming lectures. And things to think about: loss and gain of function. So most of the time, loss of function is when the gene is mutated, and the gene function is gone. So the gene can't produce the proper protein. The protein is there, but it's not active. It can't do anything. Gain of function is mostly artificially induced in a lab where you overexpress or misexpress a gene. So this means there's too much of the gene, like in the stemless, um, or it's expressed in the wrong place, which we may have a chance to talk about. And then how the rescue experiments show us or help us understand what kind of gene mutation we're dealing with. Okay, so if you take this kind of information and then look at where the gene is normally expressed, this will help you help you determine what the gene normally does. Okay. Um, yeah, and just I wanted to point this out because this is really interesting. It's quite cool. You can take a any kind of somatic tissue on a plant, and if you treat it with the appropriate hormones, you can turn it back into a complete embryo that can form a complete plant, right? So that's pretty cool, and we don't have quite the capacity to do that with animals yet. We will eventually, I'm sure, but not yet. Okay, and then just a quick summary. You can have a look at this. Next class. So I'm going to talk next time about leaves. So the decision is, where do leaves form? What is the spacing and the arrangement, and how they're shaped. And this has nothing to do with that, but it's a very, very cute picture.